Next session, I have the pleasure of introducing our next speaker, Dr. Ye Liu, who is the Senior Director of Gene Transfer Technologies at REGENXBIO Inc., a leading clinical stage biotech company seeking to improve lives through the curative potential of gene therapy. Now, Dr. Liu received his PhD in biochemistry from Wayne State University and completed his postdoctoral training with Nobel laureate Dr. Greg Semenza at the Johns Hopkins University. Prior to REGENXBIO, Dr. Liu was the Director of Genetic Engineering and Vaccine Development at Novavax. Currently, Dr. Liu is leading a team of multidisciplinary scientists to improve the safety, efficacy, and precision of in vivo gene therapy by developing novel AAV capsids and transgene cassettes. And with that, I would like to hand over the mic to Dr. Liu. Please take it away.
Thank you very much, Cole, for the nice introduction. Good afternoon, everyone. I'd like to thank the GenScript and the Organizer for inviting me to talk with you guys on this virtual summit. Hopefully, I can give you some background information and present some of our current work. So this is the first slide. My title is going to be discovering the next generation AAV gene therapy through capsid engineering and expression cassette optimization. I'm working REGENXBIO, which is a clinical stage gene therapy company located in Rockville, Maryland, just backyard of Washington, D.C. Since we are a public traded company, this is our forward-looking statement. So this is the outline of my talk today. It's going to be divided into 4 parts. In the beginning, I will spend a few minutes to explain the background of some background information about AAV gene therapy. Second, I'll show you some of our work for the capsid, AAV capsid engineering. Third, I'll explain to you how do we optimize the AAV expression cassette, some of the work we did on promoters. And last, I'll give you a case study. How do we apply this knowledge to design a novel AAV gene therapy for a rare disease called Duchenne muscular dystrophy, abbreviated as DMD. So this is a picture I took from a recent review on the history of 50-plus years history of AAV. So AAV was discovered in the 1960s by accident as a contaminant of the lab-cultured adenovirus. That's where it got the name, adeno-associated virus, abbreviated as AAV. Not too much happened for the next 10 years until the end of 70s and beginning of 1980s, and a group of scientists in the University of Florida, led by Dr. Drew Samilsky and his colleagues, first cloned and sequenced the AAV2 genome. And they go on to figure out that you can actually vectorize AAV by removing its internal genome, replace with what we call a transient. And that's how they discovered the first version of AAV vector that can be used for gene therapy. And the first human application of AAV was done in 1995 with cystic fibrosis. So of the 90s, so what people found is what we call the first generation of AAV, AAV1 through AAV6. And then some breakthrough happened during the early 2000s, 2002 to 2004. A group of scientists led by Dr. Jim Wilson and Dr. Guangping Gao at University of Pennsylvania. They sequenced a whole bunch of human tissue and non-human primate tissue, identified many more AAV capsid. Some of the names we commonly know, such as AAV8 and AAV9, AAV-RH10, they're all coming from this group of study. And this next generation AAV have shown to have improved safety and efficacy and also improved productivity. In 2008, we see the what reported by Dr. Jim Bennett again at UPenn, the first clinical efficacy of a rare inherited retinal disease called Leber amaurosis, congenital amaurosis. And then in 2012, this is the first approved per AAV product called Glabella, approved in Europe, is using AAV1. Fast forward to 2017, Luxturna, which, like I talk about this, for the treatment of LCA, and was approved by US FDA and by the sponsor Spark Therapeutics, later become Roche, and this is the AAV2 product. And then in 2019, Avexis and Novartis have their drug Zolgensma approved by the US FDA for the treatment of spinal muscular atrophy, and this is the AAV9 product. So you can see these three products ever approved, they are AAV1, AAV2, and AAV9. So this is illustration of the genome of AAV. So AAV is quite a small virus. It's what we call a non-enveloped virus. There's no lipid bilayer, for example, such as coronavirus, which has a lipid bilayer. AAV doesn't have a lipid bilayer. Its shell or capsid is a protein shell, consists of a combination of three virus protein, VP1, VP2, and VP3. AAV is quite small. The diameter is about 20 to 25 nanometer. So the packaging capacity is quite small. So what's inside of the outer shell is a single-strand DNA, which is about 4.7 kb. And this single-strand DNA are flanked by the two terminal repeat, what we call ITR, inverted terminal repeat. And in the native AAV genome, what you can see on the top of the graph, that the wild type gene has only two genes. One is replicase, another is a capsid, what we call rep and cap. And this genome is pretty smart. For example, the capsid gene. There are three proteins, VP1 to 3, but they share the same C-terminal region. VP1 have a little bit more N-terminal than VP2. VP2 have a little bit more than VP3, but they share a majority of the 3' or C-terminal sequence. And in the rep gene, they have a 3 promoter embedded in this gene, and they actually driven this P19 promoter and P70 promoter, P40 promoter. They actually driven the expression of the cap gene. And you can also see in the blue region in the middle, the intron that is control the spliceable intron control the formation of the smaller rep protein. The intron was also used by the P40 promoter to control the ratio VP1, VP3. So, also recently people identify new ORF such as AAP, and more recently another one called MAAP. So this is, this is small but clever designed virus. And then what's most importantly is Dr. Jules Milski discovered that you can basically remove all 95% of the genome. You can remove all rep and cap, replace them with what we call the transient cassette, which is in the middle of the figure that's represented by a promoter, a transient, and a poly A. So this what we call the transient cassette, flanked by the only required AAV element, which is the ITR. And then how do you make the virus? And they found that you can provide the replicase and the capsid gene and some adenovirus hypergene. You can provide that in trans, either through another plasmid transfection or through a helper virus. When you provide that, this AAV transient genome can be replicated and packaged into the recombinant AAV. Sometimes it's also called virus-like particle. So in the next slide, this is a very brief illustration of the AAV vector transduction pathway. So on the top, the AAV enter the cell through some surface receptor. A universal receptor is what we call AAVR, which is used by most type of AAV, but each AAV serotype also have their specific co-receptor which facilitate their binding to the cell surface. And then there's a process called endocytosis, and the AAV goes inside the cell through early endosome and late endosome, and during the low pH, and there's some structure change of the N-terminal, the VP1-2 region, and the AAV got released from lysosome, and after release, there's basically two paths. One is being ubiquitinized and degraded from proteasome pathway, and another pathway can actually enter the nucleus, and in the nucleus, the capsid further going through further structural change and encode to the single strand DNA genome being released, then goes through the second strand synthesis or the second strand annealing if there's more than one copy of the genome available, and then there's transcription of the mRNA, translation of the protein to have the therapeutic effect. As you can see, this is simplified version of the transduction pathway, but there's many steps that can be regulated. For example, through the binding to the surface receptor, release from the endosome, the degradation pathway, or the uncoating, the second strand synthesis. Each of these steps can be utilized through engineering or through some small molecule to improve the efficiency of AAV-mediated gene transfer. So in this slide, explain that of course, we want to maximize the full potential of AAV gene therapy. So how do I do that? At REGENXBIO, we do that through this, what we call 3 buckets. First is to improve the AAV capsid. On the figure on the left, you can see this is a novel AAV capsid called Q37. It's a cryo-EM structure that we solved with the collaboration from Dr. Jason Caliber at Rutgers University. This was published last year, and the blue core are the core region of the capsid. But outside, you see those reddish intrusion on the outside. These are what we call the variable region or the variable loop. And what happened is this variable loop are pretty tolerant to mutation or insertion, and we can actually insert up to people often do 7 mer insertion. Sometimes you can do insertion up to 10 mer, 10 amino acid, and you can add this homing peptide to increase the AAV affinity to a certain cell type. So that's one bucket. We want to improve the AAV capsid. The next is to optimize the transient. There's a promoter, there's coding sequence, there's intron, there's ITR. So we want to optimize the transient. And the third bucket on the right is what I'm not going to go into today, but equally important, it is patient-focused consideration. For example, depending on different disease indication, there could be different route of administration. Most AAV are not always giving intravenously. There could be intramuscular injection, direct injection to the brain, into the eye, etc. And the neutralizing antibody consideration, and also immune modulation for AAV, which is being increasingly noticed in the field of AAV gene therapy. So in this slide, we talk about why do we want to do capsid engineering. First, we want to seek to improve the general AAV property. And what are the general properties? For example, manufacturability. As we all know, AAV are very expensive to make, and the productivity or titer is relatively low compared to some other biologics such as monoclonal antibody. So whenever at REGENXBIO, whenever we study AAV, we want to first make sure we can produce enough it to make it commercially viable, or even produce enough for you to do the animal study. So manufacturability is the first thing we check. And second, we want to seek to improve tropism. For example, currently, the common target for AAV are generally eye, muscle, brain, liver, and sometimes the heart. Let's say improving tropism, we mean that we want the AAV to go to the tissue that we want it to go to, and don't go to the tissue that we want to detarget. We want to optimize the AAV capsid based on the particular route of administration, such as intravenous, intra-CNS, or intraocular. For example, intraocular at REGENXBIO, we are studying different routes, including subretinal and suprachoroidal. Different routes may require different capsid to even to reach a particular layer of cell types. Liver detargeting is, unless we are targeting liver for particular metabolic disease, generally we want AAV to detarget liver because liver are considered a major sink for AAV due to the vascularized structure of AAV for any vascular structure of the liver for any AAV given intravenously. And the last, there are the receptor-specific retargeting. For example, people found you can add certain binding elements such as for antibody fragment. You can link them to a certain region in the AAV capsid, and this allows us to target AAV to the particular receptor of certain cells. So how do we get new and improved AAV capsid? And first, we go through additional characterization of nature diversity. This is a real or polygenic tree of all the AAV serotype. And next, through structure-based design, like I said, there are different variable loops on the surface of AAV. One of our favorite locations is located in the variable loop 4, which records serum 455, and we see this is a exposed loop. It's pretty tolerant to all the different peptides we can insert it there. And the last is what we call the direct revolution. You can actually create a large library, millions of variants of AAV through either random peptide insertion or the IRA-prone PCR or through DNA shuffle, and we make a large library. Then we do the selection in vivo, either in mouse or in monkey. Then we harvest different organ, isolate the virus, going through multiple rounds of selection to let the nature help us to choose what is the best new serotype targeting a particular organ. So go through the first bucket of the choosing AAV through AAV natural diversity. So, like I said in the beginning, in the early 2000s, and Dr. Jim Wilson's group found out hundreds of additional AAV serotypes, including 8, 9, RH10. This whole patent portfolio of this novel AAV serotype are licensed by REGENXBIO. So we own this serotype, including 8, 9, and RH10. So, but most, you only heard a few of them, but most of them are not really studied in detail. So we want to study them. So, but we're not gonna do all 100 of them. We want to do some selection. First criteria we select is their manufacturability. So we basically, in the standard triple plasmid transfection method in HEC293 cell with a rep protein from AAV2, we only choose any capsid that can produce titer higher than 10% of AAV8. We think that's a minimal bar. It has to be above 10% of AAV8 for it even to make enough vector for our study. So after using that criteria, we choose 56, our NAV platform vector, which have all clades represented. We also choose 49 engineered vector that we made through a rational design, through a homing peptide insertion, or through a certain Y12 mutation, for example. And we also include 13 standard AAV vector. So we package this 118 different vector individually with barcoded GFP mRNA as a transgene, and after titer each individual vector, we put them together with become an AAV library, with what we call Parvate 118 library. So after we have this library, we inject them into both mouse and monkey through 3 different routes: intravenous, cerebroventricular and directly into the eye through intravitreal injection. And then we harvest different organs and isolate the DNA, RNA, and compare their through the barcode and the next-gen sequencing, compare their relative abundance. This is basically the whole next-gen sequencing workflow through initial PCR, tape station analysis, and then the Illumina MySeq sequencing. Then through the bioinformatic workflow, we're able to calculate the relative abundance of each individual barcode that representing a particular capsid, calculate their abundance. So there's very rich information in this whole data set between monkey and mouse. We can find the difference between the rodent and non-human primate. And there are some, I'm not going to go into all detail because it was presented by my colleague April Giles in AIGCT this year. So just to show a few highlights, for example, we are interested in liver. When we look at the NHP liver, a common liver capsid is AAV8 that performed pretty well and used in multiple clinical trials. We found out this new capsid called R73, which is also a clade E AAV, that the sequence homology is close to 90% compared to AAV8. So if you see, look at the graph on the left, if you look at the liver relative abundance on the DNA level, R73 is about 70% higher than AAV8. It's improvement, but not too much. But then if you look at the other peripheral organs such as heart, muscle, lung, and importantly dorsal root ganglia, which is a rich sink for AAV, you found out for example R73, it got detargeted in all the peripheral organs. And when you look at the graph on the right, the x-axis is the liver abundance, and the y-axis is the DRG abundance. So normally, so we want to choose a group of capsid that is on the lower right-hand corner, which is high liver abundance but low DRG abundance. As you can see, the first one is R73, then there's AV6 and AV3B, all share this character. And next, we are looking for the heart of NHP. Is there any better capsid than the standard AV9? AV9 is being used in multiple clinical trials for heart gene therapy. And we found out, there's a few of them, such as AV3B, Hu60, Hu12, and Hu57. They belong to either clade C or clade D. What we found out is that, again, the heart, they are, within 30–40% of AV9. They're similar to AV9. But if you look at the liver and the other peripheral organs such as muscle and again DRG importantly, and they are again very much detargeted from DRG. And if you look at the figure on the right, and you look at the lower right hand corner, these few capsids shown right there. So we can target heart but reduce in liver and DRG. I think this is certainly a favorable character compared to the commonly used AV9. Well, of course, if you want to use any of this for a particular indication, they will have to be studied more carefully in a single capsid per animal study. And this is our homing peptide study. We inserted all different homing peptides. Mostly of them are 7-mer, some of them a little bit more, like 10-mer, into this position, serine 454 in variable loop 4 of AV9. We found out some very interesting and distinct property. For example, in some of the peptides, you can actually detarget liver when you do this peptide insertion. So next, I'd like to introduce you our work on the transient cassette optimization. Commonly, workflow done are the tissue-specific or ubiquitous promoter and enhancer, depending on your indication. The intron and the splice site, and sometimes it can have consequence in the transient cassette expression level. the ITR, we can do single strand versus double strand AAV. Single strand you can utilize the full packaging capacity of AAV, but double strand sometimes can have a faster or stronger transgene expression. Protein coding regions, we commonly do codon optimization and CPG dinucleotide depletion to reduce the immune response. And there are other features such as poly A sequence, microRNA target sequence, TLR9 blocker, and certain stuffer sequence if we want the transient to be a optimal length. And finally, how you design your transient, we found out they have a lot of impact for the AAV titer, genome integrity, empty-full ratio, and potentially potency. So your product quality actually going to depend not only on the capsid, but also on the transient design. So going into our work on promoter, we are interested in design some tandem promoter. For example, this is what we called LMTB6. It's a liver muscle tandem promoter. So we know based on the work of many academic lab that liver expression of transient can be tolerogenic. So if you express a transient in the liver, it can be due to a T helper and Treg. It can be tolerated by the animals. So that's a good thing about liver expression. Also, we want muscle expression because muscle is a post-mitotic cell, and they can offer very long, potentially, a lifelong expression of a transgene. So in this case, we designed the liver muscle tandem promoter. We're using a HET promoter, a HET as a core liver promoter, a CK as a core muscle promoter from mouse creatine kinase, and then we put some enhancer sequence from the liver and the muscle. We assemble this. If you look at the in vitro data on the lower left, that we transfect the cis-plasmid in either a liver cell, QH7, or C2C12, which is a mouse muscle cell. You can see with a CAG promoter, which is a ubiquitous promoter, they are expressed, both expressed in both cell lines. The HET liver promoter only expressed in liver, and CK only expressed in the muscle, but our tandem promoter can be expressed in both cell lines. And we reproduce this result in vivo in mouse as well. If you look at the middle finger, if you look at the lower middle panel, and this is in vivo where we give the animal IV of this AAV. This is we use AAV8, by the way. And the tandem promoter offer similar expression liver as a liver promoter and higher than the CAG universal promoter. But in heart, a muscle is the tandem promoter expression level is similar to CAG and much higher than liver, showing this is a truly tandem promoter. And the same effect was happening if you do intramuscular injection as well. And next, we want to ask a question, if we do the tandem promoter, does the transcription start site still maintain similar to individual promoter? So we did this technique called rapid amplification of cDNA and then we use NGS to study the transcription start site. We found out actually, yes, both promoters start from their original start site. And which bring the question that, in this case, our 3' is a HET promoter, 5' is a CK promoter. So if the transcription start from the 5' CK promoter, then the 3' HET promoter just gonna become the untranslated region for the CK promoter. Therefore, we think we have to deplete an ATG from the second promoter, because otherwise it's gonna produce a unwanted translation initiation. So that's something we have been doing for all the tandem promoter design. And next, we analyze the intron. We have a chimeric intron in this design. We analyze whether the intron is correctly spliced out in the tandem promoter compared to individual promoter. The answer is yes. The intron is still spliced out and the canonical intron splice site. Additional to the promoter, we also want to study the enhancer. In this case, we are mining the transcriptional database to identify tissue-specific cis-regulatory module or enhancer. And in this case, you can see the upper right, the base promoter, no CRM is pretty low, and the upper middle with a native CRM is pretty strong. But with some of our designed CRM, they can achieve even higher expression level compared to the native CRM. And this apparently can be different based on either undifferentiated or differentiated cell length. So I'm going to briefly go over this. Next, we want to design some synthetic promoter rather than rely on the nature to find the promoter. In this work that was published early this year, it's a collaboration with the lab of Dr. David James at the University of Sheffield. We try to design novel synthetic promoter for controlled expression in retinal pigment epithelial cell, other than the photoreceptor cell. So the workflow, what we did is we first mining the transcriptional database to find out what are the transcriptional factor regulatory element, what TFRE, what are specific for RPE cell, and what are specific for photoreceptor PR cell. And then we try to assemble the RPE specific TFRE, put them in front of a minimal CMV promoter driving GFP expression. We use in vitro model. On the left is a cobblestone-like RPE cell that we cultured in vitro. We use plasmid transfection. The middle figure is that we use 7 copies of the same TFRE driven in front of the core CMV promoter, and we choose some of the lead from the first round selection, and then we put different TFRE in the different composition, again, check the expression level. And then this is our most recent design. What we found out is, if you compare to the very first, which is called BEST1 promoter, which is a native, very specific RPE promoter, but it's pretty weak. If you go to the very right, it's a CMV promoter, but the CMV promoter is strong, but it's non-specific. So now we have a group, we have a whole range of synthetic promoter that they can either be very specific, but strength is relatively low, or it can be less specific, but relatively strong, similar to CMV. So we have this whole range of strength and specificity ratio of synthetic promoter in our toolbox that we can use depending on the different indication. So next, I'm going to quickly go through a case study and want to leave some time for Q&A today. So this is, we use all the knowledge I explained to you, try to optimize AAV capsid and transient design in RGX-202. This is it's a product that in preclinical development currently. It's an AAV8 microdystrophin vector for the treatment of Duchenne muscular dystrophy. So dystrophin protein is located in the X chromosome. The dystrophin gene is located in the X chromosome, which encodes for a protein called dystrophin. So this is one of the largest genes in the human body, more than 4 megabase. And dystrophin is a protein involved in muscle cell structure and signaling pathway. It can bind into the protein inside of the cell at the cell membrane and transfer and absorb force within cells during muscle transfection. So in the Duchenne muscular dystrophy patient, where most of the patients are boys because it's X-linked gene. But for this patient, due to virus mutation, they don't have dystrophin. So in this case, when the muscle cell contract, there's no dystrophin to absorb the contraction energy. So this cell membrane got damaged. If you look at the picture on the right here, and so this damaged cell membrane and can lead to cell death, fibrosis, and inflammation in the muscle tissues. So over the years, and the muscle of this patient got wasted. That's what we call the muscle wasting disease. And those patients will be wheelchair bound in their teens, and some of them die due to heart or respiratory dysfunction in their late 20s and 30s. So we want to, apparently this is what we call the protein replacement therapy. The patient are missing dystrophin, we want to give them dystrophin gene. So we want, because the whole body of the patient are lacking dystrophin, so we have to use a systemic gene therapy, give the AAV intravenous so it can go to every single muscle. So the vector we choose is AAV8 because we know it can efficiently transfer gene to skeletal and heart muscle. And AAV8 has been used in multiple clinical trials. But second, there's a problem of the transient because the dystrophin is so large, even without all the introns, 11 kb, it's too large to fit into AAV. But luckily, there's a whole field of biology called microdystrophin, and people found out that you can actually only use a portion of the full-length dystrophin, and it's still functional. So we designed our own microdystrophin, and what we call the in the RGX-202, the unique feature of our microdystrophin is we have actual C-terminal, which is known to recruit key element of the dystrophin-associated protein complex to have better function. We use SPC512 promoter. It's a small and potent synthetic promoter in skeletal muscle and heart. It's discovered about 20 years ago and using very similar manner by combination of different muscle-specific TFRE, similar method as the RPE promoter that I explained to you a couple minutes ago. So there are some AAV gene therapy for DMD already in clinical trials, some of them using AAV9. One of the questions we like to ask is, how does AAV8 compare to AAV9 in non-human primate? So we got this data from our Parve 118 library study. We compare AAV8 and AAV9. We compare the DNA and RNA expression in monkeys for skeletal muscle, heart, and liver. We found out that they are really very similar. We don't see any significant difference. These are the, 3 monkeys each. So based on this data, we think AAV9 and AAV8 gonna behave very similarly in primate or human muscle after given intravenously. And this is our preclinical data. We show that we use a model of MDX it's a dystrophic mouse model, which is lacking the dystrophin protein. In a dose of 2E14 GC per kg on the figure on the left, we can achieve about 20 to 50 genome copy per cell in the muscle and heart. The middle figure shows that we can, restore the dystrophin expression in the muscle fiber of TA and diaphragm to close to 90-95% of the positive fiber. And the figure on the right to show that by Western blot, we show that our transgene expression is, this is a saturating dose, so we are getting even more than 100% of the dystrophin compared to the native level. Next, we also show the muscle function improvement through grip strength and in vitro first. Our RGX-202 gene therapy can restore muscle function to the level similar to wild type in these 2 tests. And additionally, we also studied the promoter strength. I'm not going to go into detail of this figure, but we by comparing the mRNA expression level and normalized by the DNA level, we conclude that this promoter SPC512 in muscle, if you look at the middle figure, it has a similar expression levels or similar strengths compared to the native dystrophin promoter in either in wild type or MDX mode. In heart, it actually have a stronger expression than the native dystrophin promoter. And if we compare muscle and heart, we found out that if we compare muscle and liver, we found that this SPC promoter are similar to the native dystrophin promoter. They are about 100 to 300 times more expression in muscle compared to liver. So, in summary, I show you some AAV background information, how we do capsid engineering through barcoded AAV library, and our work on the liver muscle tandem promoter with synthetic promoter construction. And finally, I introduce you RGX-202, one of the novel AAV gene therapy for Duchenne muscular dystrophy. So I'd like to thank my team of gene transfer technology at REGENXBIO. I couldn't present. This is all of their work. And lastly, I'd like to thank our Chief Scientific Officer, Olivier Danos, for his guidance and leadership. This is our picture of one of our virtual year-end meeting. And lastly, I want to put a little advertisement that we are hiring, particularly for a senior scientist working on non-coding RNA for microRNA or ribozyme, riboswitch in AAV. So if you have background in RNA biology, interest in joining REGENXBIO, please contact me. I think that will be my major presentation, and happy to answer any questions.
Thank you. Yeah, we do have a few questions and 4 minutes left, so let's get right into it. Question number one, what is the AAV titer level you're using for the DMD study?
I would assume that it means the dose level, which are in our proof of concept, we use a 2E13 GC per kg dose level.
Thank you.
In mouse study.
Is increased immunogenicity in recombinant AAVS a common occurrence?
Increased immunity, I think it depends on the dose and the route administration. We found out that when you're giving AAV high dose intravenously, there's more host immune response, but in certain immune privileged organs, such as when you're giving into the eye subretinally or giving into the CNS, there's in general less immune response.
Thank you. Yeah. And another question before we close off: does adding peptide to the capsid affect a virus stability or integrity? What kind of considerations do you take when modifying a certain AAV capsid?
I think that's a good question. I think some peptide insertion can certainly change the stability of the AAV, and also it depends on the location you insert. Commonly, people insert into for example, AAV9, we insert into the variable region 8 and variable region 4. We found out they are generally more tolerated. Also depend on the particular sequence of your peptide, and some peptide it's just not good compared to others. And second question, what kind of consideration? I think we normally try to improve tropism and targeting a particular tissue. And second is just manufacturability is a major consideration. We have to be able to produce a virus to do our study.
Thank you very much once again for your participation. We'd like to have you on our program, and I hope we see you at the next one. I will let everyone go right now as we are going to come up to the last presentation for today, which is the keynote presentation to close off the summit. So I hope to see you all there. Thank you.
All right. Thank you, Josh.
Bye-bye.
Thank you, everyone. Bye-bye.